Welcome to my channel. In this video, I'm going to explain about endospore staining technique. Aim of doing endospore staining technique is to differentiate and identify the bacterial endospores and vegetative cells from given bacterial culture. Endospores in adverse conditions like nutrient limitation, extreme temperature, dehydration. In these situations, some bacteria like Clostridium species and Bacillus species produce endospores. This endospore is metabolically inactive in its structure that are resistant to physical and chemical damages. Endospores allow the bacterium to survive in harsh conditions by protecting the genetic material of the cells. Endospores may be in spherical, subspherical, or oval in shape. They may differ in their location within the cell. That is, it may be central location, terminal location, or subterminal location, and they may or may not swell the cell. Endospores are difficult to stain with standard staining technique. Principles for endospore staining technique are: in 1922, Turner published a method for staining the endospore, but it employed a lengthy heating step. So, Schaeffer and Fulton modified Turner's method in 1933 to make the process faster. The simplest endospore staining technique is the Schaeffer-Fulton technique because of its easy and it is rapid to identify the bacteria. And it is a differential staining technique. It is used to distinguish between the vegetative cell and the endospores. Stains and decolorizer used for endospore staining technique are 0.5% of malachite green, steam, mordant and safranin. 0.5% of malachite green, which act as a primary stain, and steam, which act as a mordant. Water act as a decolorizing agent, and safranin act as a counter stain. These are all the stains and decolorizer used for endospore staining technique. Materials required for endospore staining technique are inoculation loop, microscopic slide, bacterial culture, 0.5% of malachite green dye, water, safranin, microscope and immersion oil. These are all the materials which are required for endospore staining technique. Procedure for endospore staining technique are: first, clean and dry the microscopic slide thoroughly. Prepare a smear of the culture. Next, air dry it and fix heat. Place the heat fixed bacterial slide over screened water bath and then apply the primary stain, which is malachite green. Allow the slide to sit over the steaming water bath for 5 minutes. Reapply the stain if it begins to dry out. Next, remove the slide from the water bath, cool it and rinse the slide with water until water runs clear. Next, flood the slide with a counter stain, which is safranin, for 1 minute, and then rinse it and view the specimen under oil immersion microscope. Observation and results for endospore staining technique are: endospore appears in green color. If it is vegetative cell, then it appears in red color. This is the image which shows the result for endospore staining technique. Endospore which appears in green color and vegetative cell which appears in red color. That's all about endospore staining technique. Thank you.